If you can hear me, please inform me to start. Okay, very good. Thank you. So, in the name of God, if you remember, we start and we talked about some different toxins of bacteria. Right now, we want to continue with the new one. And we go after Clostridium botulinum and Clostridium tetani. We go to the Escherichia coli. Okay. So 37 was the last page, which we told that the toxin is responsible for spasm, locked jaw, so person cannot open mouth. Maximum this match can be opened and the jaw is locked. And the muscles of the face, the muscles of the eyes, the muscles of the neck, all the muscles have spasm. The toxin can act. What do we talk about? We are talking about Clostridium tetani toxin. Toxin of Clostridium tetani, which is responsible for muscle spasm. And we explain the mechanism of action. So the toxin can act at peripheral motor nerve and plates, the brain, spinal cord, and also sympathetic nervous system. It is transported within the axon and across synaptic junctions until it reaches the central nervous system, because inhibitory inhibitory to open the mouth, cannot swallow, leading to the appearance of sardonic smile. It is like this, and leading to the appearance of sardonic smile, rhesus sardonicus. As you see, the face is look like this. And sometimes tetanus can be effective on the nervous system of the brain, so we call it cephalic tetanus, but it is rare infection involving in the middle ear that can affect cranial nerve coming from brain to the ear. And local tetanus is also rare and manifests itself as localized muscle contraction in the area of infection. So mostly it is generalized. Mostly, in most cases, tetanus is generalized, and all muscles of the body are in contraction form. But in very rare cases, maybe one part of the body has contraction, so we call it local tetanus. So it is what we discussed, and it was finished. Now we are to go through Escherichia coli. Okay. Escherichia coli. As you know, most people contain E. coli in the intestine without problem. So in most cases, E. coli is commensal. So by their presence in our intestine, we don't have any problem. But some types of E. coli, some strains of E. coli are pathogenic. Like what? Like these names: Anthrotoxygenic E. coli or ETC, Hemorrhagic E. coli EHEC, Invasive E. coli EIEC, Pathogenic E. coli EPEC, aggregative E. coli EAEC, and diffusely adhering E. coli or DAEC. But only the first two ones, Anthrotoxygenic, as its name shows, it is toxigenic. So Anthrotoxygenic E. coli produce toxin. We want to talk about this, as well as Hemorrhagic. Hemorrhagic also can produce a specific toxin, and we don't talk about the others. So from these 6 different E. coli, only we talk about Anthrotoxygenic E. coli and Hemorrhagic E. coli. So in general, E. coli is E. coli. When you do the normal test, biochemical test, PCR test, all are the same. But any species maybe have different strains, maybe have different types. So some types of E. coli are not pathogen. Some types of E. coli are pathogen. So as now all of us feel healthy, we have non-pathogenic E. coli in our intestine. So first, we want to talk about the first pathogenic E. coli as Anthrotoxygenic E. coli. What can we see in this diagram? Intestinal pathogenic E. coli, okay, from those types of E. coli which they are pathogen. Some are non-invasive, means not attacking directly to the cells, okay, not invasive, not attack to the cells. So how do produce disease? By production of toxin. So we call them toxigenic. We call them toxigenic. And because they produce toxin in enteric, in intestine, we call them Anthrotoxygenic E. coli. Anthrotoxygenic E. coli may produce one or these 2 types of enterotoxins. Some produce this one, some produce this one, and some produce both. One of them is sensitive to heat. We call it heat labile, or LT, labile toxin. And the other type is resistant to heat. The other enterotoxin is resistant to heat. So it is heat stable toxin. So labile toxin, stable toxin. First we go through these 2 ones. And in the second part, Some E. coli are invasive, attacking to intestine. From these invasive ones, some are non-toxigenic, cannot produce any toxin, but one invasive group is toxigenic, as well as attack to intestine, can produce toxin, and we call it Hemorrhagic E. coli. Hemorrhagic E. coli. And this type produce a toxin, Anthrotoxin, which this toxin is very look-like to the Shigella toxin. So it is known, it is popular with the name of Shiga Toxin. Shiga Toxin or Verotoxin. So one group, no attack to intestine, only produce two different toxins, etc. The other group attack to the intestinal cells as well as produce toxin, which its toxin is very similar, more than 60%, to the Shigella toxin. So we call it Shiga-like toxin. Sometimes there are two friends and they have two different cars. Okay, it is okay. Sometimes there are two friends, they have the same car. So what is the problem? They are completely different persons, but their cars are the same. So E. coli is completely different with Shigella, but enterohemorrhagic E. coli can produce a toxin, which its toxin is very similar to the Shigella toxin. It is what today we want to do. First we go through it, and then we go through it, and we finish our lecture today by these three ones. Okay, so first, Anthrotoxygenic. Don't forget, we are here. Non-invasive toxigenic E. coli or Anthrotoxygenic E. coli. Now we are over here. Anthrotoxygenic E. coli or ETEC is one of the most common causes of bacterial diarrheal disease in developing countries. So this E. coli come with food and water to our intestine and then produce toxin. Toxin is Anthrotoxin, and it is related to the loose diarrhea or diarrheal disease, loose motion, mostly in developing countries. Estimated 840 million cases per year annually, and in an estimated 30% of travelers to those countries with diarrheal disease. It causes acute watery diarrhea. Yes, it is water. Most in infants and adults. So in all people can make diarrhea. And it is without blood. So it is watery diarrhea without any blood. Because the inoculum of the disease is high, infections are primarily acquired through consumption of fecally contaminated food. So too much of the number of bacteria should come to intestine, and it can be achieved, it can be possible only by eating highly contaminated food or highly contaminated water, drinking water contaminated with Anthrotoxygenic E. coli. If we eat or drink which contain a low number of this bacterium, there is no disease. Immune system, normal flora suppress it. So a large number of them should be present. So person to person, talking, shaking hand, working, it is not responsible for infection transfer. Food or water should be contaminated. For example, vegetables or water should be highly contaminated with this bacterium. And by eating, this problem will be started. Okay. Yes, thank you, Mr. Ali Majid Abbas. So next week we go through the Shigella and we discuss regarding how is the toxin of Shigella and how can we understand related that. Okay. Common serotypes associated are, so they have different serotypes. There is no need for memorizing. So knowing that we have Anthrotoxygenic, and Anthrotoxygenic have different types. They are in different types. And all of these serotypes and all of Anthrotoxygenic E. coli, they are toxigenic. They can produce toxin, but they are not invasive. It is what? There is no blood. Because bacterium don't invade to the intestine, only produce toxin. Only producing toxin. Pathogenesis of Anthrotoxygenic E. coli involves attachment. So when they come with water or they come with food to intestine, first ETEC, Anthrotoxygenic E. coli, attach to the intestinal mucosa. How? By their Fimbria protein. By use of their Fimbria proteins. What do we call them? We call this Fimbria in Anthrotoxygenic E. coli as colonization factor antigen, CFA. Okay, so here you can see E. coli, if come to the lumen of intestine, come very close contact with microvilli of the intestine, and the bacteria have pili or Fimbria with the name of CFA pili, and by this pili attach to microvilli and then start production Anthrotoxin, then starting for production of Anthrotoxin. What was the CFA abbreviation for? CFA, colonization factor antigen. What is that? It is fimbrial protein which ETEC by use of them can attach to the microvilli of the intestine and then start toxin production. Some toxins are heat stable and some toxins are heat labile. Some are heat resistant, some are heat sensitive. ETEC produced two classes of Anthrotoxins, heat stable or ST and heat labile LT. So they can produce ST and LT. Two different toxins can be produced. Okay. Yes, absolutely, it is correct. Hemorrhagic E. coli produce verotoxin. Yes, I think we received some of your message by delay. So you talk about this part, okay? But now we are in this part. So when we finish Anthrotoxygenic E. coli, LT and ST, then we going to Anthrotoxygenic E. coli and talking from this. Okay, I think when I was explaining this part, you send your message. So after 30-40 minutes, after finish this part, we will come to discuss about Shiga-like toxin or verotoxin as you wrote. So heat-stable toxin, STA, but not STB, is associated with human disease. So E. coli produce STA. This STA is the causative agent for disease in human. But STB, no, it is not associated with human disease. Is found in 75 to 80% of ETEC. So most strains, most Anthrotoxygenic E. coli produce this one, ST. Okay? Stable toxin, resistant toxin to heat. Along or associated with LT, and some of them can produce also sensitive toxin to heat or labile toxin, and is responsible for commonly for severe disease than LT only. So if one bacterium only produce this one is weaker, then they produce ST or ST along with LT. Secretory diarrhea caused by ETEC. So the diarrhea caused by ETEC develops after 1 to 2 days incubation period and persists for average 3 to 5 days. So incubation period is 1 to 2 days, and person disease continues for 3 to 5 days. The symptoms: watery, non-bloody diarrhea, and abdominal cramps. There are too much gases produced in the intestine. Less commonly nausea and vomiting are similar to those of cholera. So the symptoms look like cholera. The causative agent is Vibrio cholera, but are usually milder, although mortality is high in malnourished individuals, so those infected people with bad nutrition, and in those with underlying disease, particularly children and elderly, are in risk. They are in high risk. Diagnosis is done by detection of toxins by in vitro and in vivo. So here we don't want to talk about identification and going to the bacteriology. We want to focus only on toxin. Yes, our responsibility in this course is thinking and talking about toxins, microbial toxins, and their mechanisms of action. So we came to this part that we have in some people diarrhea. The causative agent is Anthrotoxygenic E. coli, and this ETEC can produce heat-stable toxin or ST, heat-labile toxin or LT. When this bacterium with water or food come to the intestine, adhere to the epithelium of small intestine. Yes, they adhere by CFA, as I explained you. And the presence of bacteria with production of Anthrotoxin is responsible for nausea, vomiting, and loose stool. LT, labile toxin, sensitive toxin, is look like cholera toxin. So next week we go through cholera toxin and we explain the similarities. Cause accumulation of fluids. What happened? So bacteria with CFA attach, produce Anthrotoxin. Anthrotoxin come into the cell, intestinal cells, and effects on cyclic nucleotides like cyclic adenosine monophosphate, cyclic guanosine monophosphate, and other mediators. So the ions of color, sodium, potassium, carbonate, and water excreted outside to the lumen and going out with feces, going out. So it is diarrhea. What is the meaning of diarrhea? So different ions from cells excreted to the lumen of intestine and with diarrhea go out of the body. So cause accumulation of fluids. So there are too much water. So it is watery stool. And they go out from the body. This CFA or Fimbriae or Pili is a virulence factor, which accordingly this bacteria can attach to the intestinal cells. So adhesive factors or Fimbriae, they are specific, they attach to specific receptors in intestinal epithelium. Most mortality can be seen in children lower than 5 years. Okay, so the other thing we want to know that which genes are responsible for toxin production in ETEC? Is it related to the chromosome or plasmid? No, it is not related to both of them. It is not related to the chromosome. It is related to the plasmid. Plasmid mediated. So these two toxins, ST and LT, are related to the plasmid genes that stimulate hyper secretion of fluids and electrolytes. So again, as you see, it is intestinal lumen, it is intestinal epithelium. Intestinal epithelium have receptor for pili, and this pili we called as CF or CFA. Accordingly, they attach to the intestinal wall. We call it colonization. After completion of colonization, production of sensitive toxin to heat and resistant toxin to heat, LT and ST, are started, and these toxins going inside cell, and they are causative for stimulation, hyper secretion of fluids and electrolytes. What is the diarrhea? Diarrhea is hyper secretion of fluids and electrolyte. So don't forget that these toxins are related to the plasmid, extrachromosomal DNA, and they are not related to the chromosome. They are not related to the chromosome. ETEC strains cause diarrhea, many times repeated and repeated. They are caused for diarrhea through the action of enterotoxins LT and ST. These strains may explain an LT only, ST only, or both LT and ST. So some of the ETEC produce only one LT or one ST, and some strains can produce both of them. Disease requires bacterial attachment. Yes, we can see it is attachment to the small intestine epithelium. Yes, we can see by the bacterial surface proteins. Yes, they are surface proteins, and we call them colonization factor. It is CF, and elaboration of heat stable and heat labile enterotoxin, then starting for production of enterotoxin. And you can see in different parts of the world, mostly in warm places, this type of E. coli is prevalent, and they are causative agent for most diarrhea. So again, reminding the genes which they should be expressed are related to the plasmid of the ETEC. The genes for colonization factors and enterotoxins are encoded on a transmissible plasmid. So if one ETEC contained plasmid can transfer the copy of the plasmid to the another same type with absence of plasmid. So both of them can have it. And again, if ETEC contain plasmid, can produce ST or LT or both of them. Heat stable toxin, STA, is only associated with human disease. Yes. STB now. STB no. Only STA is found in 75%, 80% of ETEC, either alone or associated with LT, and responsible for more commonly for severe disease than LT only. So this one is causative agent for more severe disease than this one. If two person, one E. coli containing ST, and in another person E. coli contain LT, so the severity of disease in ST containing E. coli is more than the other one. Now we want to understand what is the ST mechanism of action. ST mechanism of action. We want to go through it. ST means heat stable toxin. This first one, which is more dangerous. Yes, STA is a small monomeric peptide that binds to the transmembrane guanylate cyclase C receptor. So this toxin finds a special receptor which is related to the guanylate cyclase. Okay? By activation of this receptor on the intestinal epithelium, leading to increase in cyclic guanosine monophosphate. So when this toxin STA attached to this receptor inside the cell, there are too much cyclic GMP or cyclic guanosine monophosphate. The result of presence too much cyclic GMP subsequent is hyper secretion of fluids as well as inhibition of fluid absorption. It is induction for fluids going out of the cell to the lumen and not allowing new fluid coming back. We can see the photo over here. Heat stable toxin, it is ST. We told that when the bacterium attached to the intestinal microvilli produce ST attached to the receptor, and then receptor guanylate cyclase activate this enzyme. This enzyme is responsible for high number of cyclic GMP in the cell, and with this not allowing water and ions come to the cell and inducing water going out to the lumen. Okay, this is the mechanism of action of ST or heat stable toxin. STA is a small monomeric peptide, so it is a protein that binds to the transmembrane guanylate cyclase C receptor, okay, attached to this receptor. What is this receptor? This is related to the enzyme guanylate cyclase. What is the result? The result is leading to the increase of cyclic GMP. What is the result of presence too much cyclic GMP? Subsequent hypersecretion of fluids as well as not allow its fluid coming back or absorption. It is the mechanism of action of ST. Now, what is the mechanism of LT? What is the mechanism of action of LT? Is the toxin which is sensitive to heat. So, labile toxin, LT, LT1 is more commonly associated with human disease. So LT1 is functionally and structurally similar to cholera toxin. So this toxin LT in ETEC, according to the function and according to the structure, is very similar to the cholera toxin. So there is no problem. Like two persons have the same car. Both, not one car. One person has one car and the other person has another car, but those two cars are similar. So the toxin of LT is very similar to cholera toxin according function and structure. How much homology does it have? 80%. Homology can be observed and consists of one A subunit and 5 identical B subunits. So it has 2 subunits, A and B, 5 identical B subunit and 1 A subunit. Those B1s are effective for attachment and helping a subunit come inside the cytoplasm of intestinal cells. So B is working, B subunits are working, look like taxi, look like shuttle, look like carrier, and the important part is the A site. So B subunits attach to the cell and helping A subunit come inside the cell, then dissociate with each other, B subunits going out and A subunit remaining inside the cell. The B subunits bind to the same receptor as cholera toxin. We call these receptors as GM1, gangliosides. So in intestine, there are some receptors with the name of GM1 gangliosides. They are specific for Vibrio cholera toxin and LT toxin of E. coli, as well as other surface glycoproteins on epithelial cells in the small intestine. After endocytosis, so the toxin completely by endocytosis come inside the cytoplasm. The A subunit move across the membrane of the vacuole and interacts with the membrane protein GS that regulates the adenylate cyclase. Okay, don't forget that previous toxin was activating the guanylate cyclase, but LT activate adenylate cyclase. ST activated guanylate cyclase with production to match cyclic GMP, but LT activate adenylate cyclase with production to match cyclic adenosine monophosphate. But the result of both of them are the same. The result of production to match cyclic AMP is to match secretion, water, and ions to the lumen. Too much water and ions going to the lumen, and it is diarrhea without any blood, without any. So these two ones here, you can see they are very, very similar with small differences, So they can activate adenylate cyclase. The net effect of this interaction is an increase in cyclic adenosine monophosphate CAMP levels, resulting in enhanced secretion of chloride and decreased absorption of sodium and chloride. These changes are manifested. These changes are resulting watery diarrhea without blood. Exposure to the toxin also stimulates another things like prostaglandin secretion and production of inflammatory cytokines, resulting elevation of further fluid loss. So here you can easily see two-page explanation regarding the mechanism of action of LT and ST of Anthrotoxygenic E. coli. LT is effective on adenylate cyclase. Adenylate cyclase overproduce cyclic AMP, and cyclic AMP overproduction in the cells is responsible for secretion water and ions, and ST is responsible for activation of guanylate cyclase, and overproduction of cyclic GMP is responsible for secretion water and ions and not allowing absorbing water and ions. Okay, so as I told you, this part finished. Okay, we discussed about mechanism of action of LT and ST. Now we are in this part. Now we are talking about intestinal pathogenic E. coli, which they are invasive, attacking to the intestine with blood production as well as toxin production, and their toxins are very look like to Shigella toxin. We are here. Yes, we are here. Hemorrhagic E. coli. So easily we can understand so this bacteria place of action is intestine, and hemorrhagic means production of blood, means bleeding of intestine, bleeding. And E. coli, which we are talking about E. coli, pathogenic E. coli, EHEC. But this bacterium have another names also. It has another names also. Like what? Because their toxin, because EHEC toxin is very look like to Shigella Toxin. Sometimes we call it Shiga Toxin producing E. coli. Shiga Toxin producing E. coli. So it is E. coli which producing a toxin which is look like the toxin of Shigella. Shiga Toxin producing E. coli or STEC. So EHEC and STEC are the same. And sometimes we call it Verotoxin producing E. coli, VTEC. So EHEC, STEC, VTEC, all are one. All are the same names of one thing. They are not different. Because this toxin is very active on tissue culture containing vero cells. Sometimes we call it verotoxin producing E. coli. All strains of Hemorrhagic E. coli produce Shiga toxins that destroys vero cells. I told it is a tissue culture. Similarly to Shiga toxins produced by Shigella. So the toxin of Shigella and the toxin of Hemorrhagic E. coli are the same. There is no problem, yes? Two bacteria have very close related toxins. E. coli O157H7, it is serotype, yes? Because we are talking about E. coli. As you do PCR, they are E. coli. If you do biochemical test, they are E. coli. But according serology, we can understand their types. We call it serotyping. We call it serotyping. So H is related to the antigen of flagella, and O is somatic antigen is related to the antigens of cell wall. You know, E. coli is a gram-negative bacteria. So O157 H7 shows the serotype of Anthrotoxygenic E. coli. So if we have 100 E. coli, all are E. coli, but which of them is Anthrotoxygenic? We can do serotyping. By serotyping, serotype of O157 H7 is the Anthrotoxygenic E. coli, is primarily cause of EHEC infection in industrialized countries including USA, Canada, and England. Based on the severity of the disease, Anthrotoxygenic E. coli is regarded as the most serious E. coli strain among foodborne pathogens. Among all bacteria which can come to our body and make food poisoning, this strain has the most severity. E. coli O57H7 differ from other E. coli serotype because of some of its characteristics, which are hemorrhagic E. coli not being able to grow in or above 42 degrees centigrade. So most other E. coli can grow on 42 degrees centigrade, but none of hemorrhagic E. coli can grow. Not being able to ferment sorbitol, but the others mostly can use sorbitol. Not having beta glucuronidase enzyme. This is a disability of production of this enzyme and producing enterohemolysin. Enterohemolysin can produce, the others cannot. This toxin, which is very look-like to the Shigella toxin, produced by this serotype, is cytotoxic. It is toxic for cells, any cells which they can reach, mostly to the intestine, mostly human colon. This is a part of intestine, and first part of intestine after stomach is duodenum. First part of small intestine after stomach, it is duodenum. This toxin causes accumulation of fluid in intestine, so it is diarrhea, and lesions, it is blood in colon through destruction of crypt epithelia. So there are some crypts in the epithelial cells of intestine, and by destruction, because it is invasive, there are many blood. Intimin, it is a protein using, makes adhesion to the intestinal canal easier. So by this can easily attach to the intestine. EHEC has a wide spectrum including watery or bloody diarrhea. Sometimes it is only watery, and most of the times diarrhea is bloody. And hemorrhagic uremic syndrome. What is this? Syndrome means disease. Uremic means related to the urea, related to the urine. And hemorrhagic means blood. So sometimes this bacterium attack to the urinary system and in the urine of the person there are many blood and the urine color is red. What do we call it? We call it hemolytic uremic syndrome. Hemolytic uremic syndrome, which is an important factor in acute renal failure in children. So this bacterium in children can attack to the kidney and be causative agent for renal failure. Kidney not working properly. Kidney cannot filtrate the blood appropriately. So this toxin, yes, related Shigella toxin, this STX is essentially identical to Shigella toxin, many times repeating. It is look like Shigella toxin, which produced by Shigella dysentery. The source of the name, accordingly, it has this name. STX2 has 60% homology with XTX1, and both toxins are acquired by lysogenic bacteriophages. What is that? So if you come back to the previous lectures, some related genes to toxins were related to the chromosome. Some toxin productions were related to the plasmid. But here in Anthrotoxygenic E. coli, the genes responsible of toxin production are related to the bacteriophage. They are related to the lysogenic bacteriophage. What are they? Sometimes some viruses, some bacteriophage attach to the bacterium and then injecting the viral genome to the cell. So this is DNA of virus, this is DNA of bacterium, and then DNA of virus incorporate to the DNA of bacteria. So the blue one is the chromosome of bacterium. Bacterium is E. coli. And the red one is prophage of virus. The prophage of virus. Then this virus is responsible for toxin production, and E. coli is more powerful in attacking to the intestine by help of this toxin. So this virus is benefit for bacterium, and bacterium allow this virus multiply with bacteria. When bacterium divide to 2 bacteria, so we have 2 blue and 2 red. So it is multiplication of virus without any problem. Then we have 4, 8, 16, 32, 64, 128 bacteria, and all of them have bacterial chromosome as well as virus, and in all of them virus is responsible for Shiga toxin production. For Shiga toxin production. So for a long time This is positive, positive. Bacteria help to virus, virus help to the bacteria. But sometimes, after many cycles, bacteria going out from lysogenic cycle to the lytic cycle. In this, virus immediately multiply and break down the different parts of the bacterium and then break the bacterium and run out. They come out. We call it lytic cycle. So here what did we wrote? We wrote both toxins STX1, STX2 are acquired by Anthrotoxygenic E. coli by coming lysogenic bacteriophage to the bacterium, and we have prophage. What do we have? We have prophage, the incorporated bacterial genome to the genome of the bacterium, the incorporated of viral genome to the bacterial chromosome we call prophage. So when there are prophage of virus to hemorrhagic E. coli chromosome, this bacterium can produce Anthrotoxin and can have invasiveness with bleeding and attacking to the intestinal cells, accompanying with diarrhea, bloody diarrhea. So both of these toxins, STX1 and 2, both of these toxins have two subunits, A subunit and B subunit. And one complex is containing one A subunit with 5 B subunits. As I told you, B subunits are working, look like a taxi, like a shuttle, like a carrier, and take A subunit to the cell. So with the B subunits binding to a specific glycolipid on the host cell, and we call it GB3, infection starts. A high concentration of GB3 receptors is found in intestinal villi and renal epithelial cells. So now we understand why this toxin, like intestine and kidney, because these receptors are present in these two parts. GB3 are present in intestine and kidney. So this toxin is effective on intestine and kidney. After the A subunit is internalized, by help of B subunit to the cells of kidney and intestine, it is cleaved into two molecules. It is cleaved from B subunits and it is cleaved from itself. So it has two parts, A1 and A2. So A1 is very important fragment. Bind to 28S ribosomal RNA and cause a cessation of protein synthesis, not allowing protein synthesis. Protein synthesis of these cells are stopped. STEC strain with both Shiga toxin and attaching and affecting activity are more pathogenic than strains producing only one Shiga toxin. Hemorrhagic Rhemic syndrome, when they attack the kidney, has been preferentially associated with the production of STX2, which has been shown to destroy glomerular endothelial cells. So when glomerular endothelial cells are destructed, filtration is not working properly in the kidney. Damage to endothelial cells leading to platelet activation and thrombin deposition, which results in decreased glomerular filtration, so not good filtration in kidney, and acute renal failure. Acute renal failure means stop working of the kidney. The Shiga toxins also stimulate the expression of inflammatory cytokines like tumor necrosis factor, interleukin 6, and enhancing expression of B subunit receptor GB3. And last page. Now we see the mechanism of action. What did we talk? Mechanism of action of Shiga toxin and the toxin of enterohemorrhagic E. coli. Binding of the bacterium to the GB3 layer of the cell. Yes, this is binding to the GB3. 2. Production of Shiga toxin. Yes, it is production Shiga toxin. 3. STX is transported into Golgi complex. These toxins going to the Golgi, how they go there? Endocytosis of the toxin in the cell. It is endocytosis. Toxin is coming to the cell by endocytosis. Clathrin enveloped vesicle formation. So vesicle is formed with some protein, clathrin. 6. The toxin is transported to the Golgi complex. The toxin going to the Golgi complex. Okay. 7. Vesicle breaking and separation of the pentamer B from the toxic A fraction. So these 5 B subunits going out and only A1 remaining, this yellow one. Only A1 is remaining. Action of the A1 portion on the ribosomal RNA in 28S protein acting as N-glycosidase, replacing an adenine moiety and inhibitory to the translation of RNA tape. So with stop translation, with stop ribosome, all ribosomes work, cell will die. And with die of many cells of the intestine, many cells of the kidney, the disease is produced. Okay, it is sufficient. So next week we go to the Shiga toxin. As you know, Shiga toxin is completely look like to enterohemorrhagic E. coli toxin, and mechanism of action is like this, which now I told you. So next week we go to the Shigella or Shiga toxin, and we discuss about it and we go forward. If it is any question, please ask me. Please have some review during the weeks remaining to the end of the semester. If you find any problem, please ask me. And some toxins are completely different with each other, and some toxins have many similarities with another toxins. It is what we should explain today, so it is finished. Thank you because of your presence. So right now 20 students are present. I'm happy. So if any question asked, if not, we will meet each other next week at the same time in the same place. Thank you very much. Goodbye. See you. Have a good time.